Hello class, this is Dr. Anderson and I will be responsible for teaching endocrinology this semester, and so we're going to start with some cellular basics of endocrine physiology. So we always start with learning objectives. The first learning objective is a large one. I want you to compare and contrast steroid hormones versus protein hormones. Now there can be many categories of hormones, so glycoproteins and peptides, amino acid derivatives, steroids, eicosanoids, all those sorts of things. But I'm going to ask you to sort of boil down the categories into two. So lipid-soluble steroids versus water-soluble protein hormones. And here are the things I want you to think about as you're doing that comparison. What is the chemistry of these substances? For example, steroids are cholesterol derivatives, whereas proteins are made from amino acids. The location of their receptors. Are their receptors inside the cell or in the plasma membrane? How are they carried in the blood? Do they use a carrier protein, or are they free in the blood? What is their half-life? How long does this hormone stay around in body systems? And finally, when they bind to their receptors, what kind of intracellular mechanisms carry out the will of that particular hormone? And a way to achieve that learning objective is to work through Critical Thinking 8.1. Next, we want to talk about something called upregulation and downregulation. So this learning objective gets at what is the relationship between the receptor number and the tissue sensitivity for a given hormone. So this isn't about changing the affinity of a receptor, making a receptor from high affinity to low affinity. Nothing changes in an individual receptor. What happens if you have more receptors or if you have fewer receptors? What happens in terms of tissue sensitivity and tissue response? Finally, I want to introduce a concept called permissiveness. And I'd like you to be able to recognize examples of permissiveness and be able to discuss reasons why a hormone would be permissive. And then you would be able to achieve that outcome through working through Critical Thinking 8.3. So here's a picture from your textbook showing a person and all the various organ systems. So we will be talking about the hypothalamus and the interaction between the pituitary, and we'll divide the pituitary into the anterior and posterior pituitary. We'll talk about the regulation of the thyroid gland, not so much the function. We will talk about, well, right on top of here, the regulation of the adrenal gland. Actually, later on in the semester, we'll talk about the regulation of the pancreas. After we finish endocrine physiology, we'll talk about reproductive physiology, and we'll talk about male reproduction with regulation of the testes and female reproduction with the regulation of the ovaries. So endocrine glands are glands that secrete hormones. And we define a hormone as a chemical substance secreted into body fluids, particularly the blood. And a gland could be one cell or a group of cells, and the secretions of those cells then have a physiologic control effect on other cells of the body, and they do so through a specific receptor. So when we talk about this in class, and as you're viewing this presentation now, think to yourself, what are some examples of hormones that you know? Now, hormones need to work through receptors. The idea of a receptor is key to a hormone. So here's an endocrine cell. It's releasing its hormone product. That hormone product goes into the blood. Doesn't have to be the blood, could be body tissues, but most of the hormones we'll be talking about go through the blood. Go through the blood, leave the capillary system. Now, if that hormone finds the right receptor, it will have an action on that cell, that target cell or the effector. Notice there are other cells with other receptors, but those receptors don't bind that hormone. So that hormone has no way to affect this cell because there is no hormone. But we have a cell here and we have a cell here. Those are the targeted cells because they have a specific receptor that is able to bind and react to that particular hormone. So endocrine glands, this is a picture from a different textbook, but it shows the concept very nicely, that endocrine glands produce a product that goes into the blood. This is very different than another kind of gland called an exocrine gland. Exocrine glands have ducts, and their secretions go out upon an epithelial layer. So for example, the salivary glands are an example of an exocrine gland, whereas the adrenal gland is an example of an endocrine gland. So what do we know about hormones? Actually, you find very, very tiny concentrations in the blood because that small amount of hormone ends up being quite potent. And hormones are very potent because they engage in a process called amplification, where one hormone binds to a receptor, and then beyond the receptor, the response of the cell gets bigger and bigger and bigger. Hormones regulate biologic functions. So they regulate blood glucose level, they regulate metabolism, they regulate a stress response, they regulate reproductive function. And they do that through receptors that are specific. So, for example, estrogens are a type of hormone that bind to estrogen receptors. Now, there are subtypes of estrogens, estradiol, estriol, or estrone, but they all are estrogens and they bind to an estrogen receptor. Estrogen is not going to bind to an insulin receptor or to an FSH receptor, because the ligand receptor interaction is very specific. So we're going to start with the lipid-soluble hormones. The lipid-soluble hormones are mostly the steroids. That's where we will direct our conversation. Steroids are cholesterol derivatives. So cholesterol has 4 little organic rings with some side chains. Because steroids are lipid-soluble, they don't dissolve in the blood very well. They're not very water soluble. So they rely on a carrier protein that carries it through the blood. And the interesting thing about the carrier protein is that that protein protects the steroid from being broken down rapidly. And therefore, lipid-soluble hormones such as steroids tend to have longer half-lives. So if we look at this diagram, here's well, at the bottom, we'll start here, this kind of purplish color. This is a molecule of cholesterol, and you see there are 4 rings with some side chains. Now, here are 4 examples of steroids. Cortisol, do you see the 4 rings? But the side chains are different. Aldosterone, again, we have the 4 rings, but it's the side chains that are different. Testosterone, again, that same basic pattern. And estradiol with its different side change. Notice that there's a lot of similarity between estradiol and testosterone. This side group is changed, and that change is brought about by a hormone or by an enzyme called aromatase. We'll talk about that later. Anyway, I want you to see the general pattern of what a steroid looks like. So steroids are synthesized by specific cells, glandular cells, and that cell has to get some kind of signal, could be a different hormone, and turns on processes inside the cell. Cholesterol has to be harvested from LDL, which are low-density lipoproteins. That's one way cholesterol is carried around in the blood. And through the action of enzymes in the mitochondria and the smooth endoplasmic reticulum, they work upon the cholesterol side chains and form a steroid hormone. And because steroids are lipid-soluble, they can diffuse out of the cell. So what we see in this side picture, cholesterol can be converted into all sorts of hormones. Notice corticosterone. That's the cortisol that's produced in mice and rats. Corticosterone can be turned into aldosterone. There's progesterone. We have another intermediary that can be turned into cortisol. Or these are male androgens. So they share biosynthesis pathways. So cholesterol can be converted into this androgen precursor and acted upon by aromatase and turned into estrone, and then estrone can be turned into estradiol. Or the male steroid can be turned into testosterone, which can be acted upon by aromatase and then can get converted into estradiol. So the point of me showing you this is not for you to memorize a bunch of biochemical pathways. I want you to see the idea that if a man took some excess testosterone, that some of that testosterone would be converted into estrogen. And if he was perhaps taking anabolic steroids to bulk up his muscles, that's not something that I would advocate that people do, but you've read about it in the news. If someone's taking a steroid to bulk up their muscles, they also have to take an aromatase inhibitor to prevent the production of estradiol. So if you are an endocrinologist and there's an excess in steroid, there will be excesses in other steroids because they share biochemical pathways, and that makes the endocrinologist's job very challenging to figure out what is the primary disorder that then is affecting the synthesis of other steroids. So let's stop and think about something. You have learned about neurons from Dr. K. You've learned about alpha motor neurons from Dr. Barnett. Neurons make neurotransmitter, package them in vesicles where they sit at the end of an axon terminus until they get the signal to be released. Why aren't steroid hormones packaged within secretory vessels and stored within the cell? So give yourself a chance to think about that. Well, we know that steroids are lipid soluble, and secretory vesicles are made out of phospholipids. So the steroid wouldn't stay there. It wouldn't be an effective way of storing steroid hormone. They would diffuse out of the vesicles. So that's why we don't store them in vesicles. They wouldn't stay there. So because steroids are lipid soluble, instead of storing steroid in the cell, we basically store it out in the plasma, bound to protein carriers. Some carriers are very specific. So there's androgen binding globulin, or sex hormone binding globulin, or thyroid binding globulin, or glucocorticoid binding globulin. So they're very specific for various receptors. But some are general carriers. Albumin is a very large protein found in the blood. Albumin exerts something called protein osmotic pressure, or oncotic pressure. That's one of the Starling's forces. Another job that albumin has is that it carries things around in the blood. Note, though, that the hormone has to unbind from its carrier, diffuse into a cell to have its biological action. So the free hormone is the one that's biologically active. So this is a picture from your textbook showing you we have a steroid hormone, and notice it's being carried in a protein carrier. That allows it to dissolve in the plasma. That also allows it to extend its half-life. Water-soluble hormones are easily able to dissolve into the blood, and so they don't need a carrier. So most of the steroid is bound to a carrier protein, and since only the free hormone can diffuse into the liver and be broken down, The carrier is shielding the hormone from breakdown. As a matter of fact, the more lipid-soluble a hormone is, the greater it binds to its carrier, and the more it's shielded from degradation, and that extends the half-life. In fact, you could plot lipid solubility versus half-life, and you'd get a fairly linear relationship. So, for example, the half-life of estradiol is 13 hours. The half-life of water-soluble hormones are maybe a few minutes. Now this diagram shows you how a steroid hormone is going to work. So here's our lipid-soluble hormone. It could be testosterone or estradiol or any number of steroids. They diffuse into the cell. Diffuse into the cell. They can do that. They can diffuse across that lipid bilayer. And inside the cell, they're going to find its receptor. So notice in this particular case, the steroid and the two receptors form a dimer, and now you have the hormone and the dimer, and they together act like a transcription factor. They bind to a particular part of the DNA. That DNA is transcribed through the action of RNA polymerase. That messenger RNA then goes out into the cell and is made into a protein with the help of ribosomes. So you have transcription, translation, and then the action of the protein. So we have gene expression, or the synthesis of a protein that then causes some kind of cellular response. And that's basically how steroids work. So why are steroids described as slow-acting but long-lasting? Well, number one, steroids work through gene transcription. Gene transcription takes some time. You have to make brand new proteins, fold those proteins up, activate them, and then the proteins go out and carry out the function of the hormone. That takes some time. They're also long-lasting because they have a long half-life. Alrighty, let's go on to peptides, proteins, and glycoproteins. We all sort of put that together in one big category. These are larger molecules. They dissolve very well in blood, and they're broken down by peripheral enzyme systems. Sometimes that's out in the blood, sometimes that's inside the cell, and people are basically studying how peripheral enzymes break down peptide hormones. So for example, the chemical half-life of growth hormone is 20 to 30 minutes. Do you remember what the half-life of estradiol was? It was 13 hours. Growth hormone is going to be around 20-30 minutes. Actually, it's more accurate to say in 20 to 30 minutes, the concentration or the amount of growth hormone is going to be cut in half. That's why we call it a half-life. Now look at epinephrine. Epinephrine is an amino acid derivative, and so it's much smaller than your typical protein hormone, but it acts like the protein hormones because it binds to an extracellular receptor. But notice its half-life, 2 minutes. In 2 minutes, half of your epinephrine has been broken down. So water-soluble proteins and peptides have much shorter half-lives. When they bind to their cells, they tend to work through a second messenger system. So for example, this is cyclic AMP. So we'll have a hormone, could be ADH, antidiuretic hormone, binds to the receptor. The receptor is aligned with a heterotrimeric G protein. Binding activates the G protein, and the G protein unbinds, and then you get the G protein comes apart and activates a membrane enzyme called adenylyl cyclase, which converts ATP to cyclic AMP. Cyclic AMP then activates a protein kinase A. Protein kinase A then phosphorylates proteins and either turns them on or turns them off. So many hormone systems work through a cyclic AMP system. Now once you have the second messenger like cyclic AMP, you can turn on all sorts of things. You can activate a transporter. You could open or close an ion channel. You could release calcium. You could turn on an enzyme or turn off an enzyme. You can have organelles moving along microtubules. Or the second messenger could turn on a transcription factor that then works on DNA synthesis and RNA synthesis. So a second messenger can turn on a protein, and that could be very fast. But a protein hormone could have lots and lots of long-term effects through gene transcription. So if something happens in a matter of minutes, it's probably working through a second messenger. If it takes hours to see the effect, it's probably working through gene transcription. Now, why should we use a second messenger system? They're kind of complicated. Why do we do all of that stuff? Because it gets us something called amplification. Here's our first messenger and our receptor, and we produce some cyclic AMP molecules that then turn on a bunch of protein kinases, which then phosphorylate a bunch of enzymes, and those enzymes produce a bunch of product, and we can go from one molecule to a million molecules through one pathway. So that's a big increase in response. And that's why we don't have to have very much hormone hanging around in the blood, because hormones are very potent through this process of amplification. So thinking about what we've learned so far, why would we say proteins are fast-acting but short-lasting? Well, they're fast-acting because they work through second messengers and turn on proteins that are already present within the cell. And they're short-lasting because they are not shielded from degradation. They have ready access to their breakdown processes, and they have very short half-lives. Now, what about this hormone versus receptor level? Because certainly, if you wanted to have a hormone effect, you could secrete more hormone. But that's not the only way to regulate a hormone response. You can also influence the sensitivity of a tissue for the hormone in question through changes in receptor number. So this is a picture from your textbook showing that we've got a receptor with 4 little membrane receptors. If those receptors are decreased in number, here they go from 2 and down to 1, over time we have downregulation, and this target cell is less sensitive to the ligand. In our second example, we have 1 receptor, then we have 2, then we have 4. Over time, as receptor number is increasing, this target tissue is more sensitive. So, for example, if a person is chronically stressed, they will downregulate their growth hormone receptors. Then the target cells are less sensitive to growth hormone. In the presence of thyroid hormone, we upregulate beta receptors in the heart. So as those receptor numbers increase, we have a greater effect of epinephrine through beta receptors on the heart. So downregulation decreases sensitivity, upregulation increases sensitivity. So if we increase the receptor number, we increase tissue sensitivity. So if we upregulate beta adrenoreceptors in response to thyroid hormone, the heart is more sensitive to catecholamines in the presence of thyroid hormone. So you could increase thyroid hormone and get an increase in heart rate without increasing epinephrine. Likewise, with downregulation, we're decreasing the receptor number, we're decreasing tissue sensitivity, and if someone is chronically stressed, they could decrease their growth hormone receptors. And let's face it, if you are trying to deal with stress, your primary function, or the primary thing you're trying to do, is survive. Growth is less of a priority. So let's do this little critical thinking exercise. Oxytocin is a hormone that causes uterine contractions. If the oxytocin levels stay the same, but oxytocin receptors upregulate, what will happen to the smooth muscle tension in the uterus? Well, I hope you realize even if oxytocin doesn't increase, target tissue sensitivity to oxytocin will increase, it'll have a greater effect, and the uterus will start to contract. As a matter of fact, we think that's the way labor starts in humans, that the oxytocin receptors upregulate, uterine contractions start, and then oxytocin levels increase secondarily to the upregulation. All right, how about how hormones interact? So I have 3 graphs here that show 3 different effects. Hormone A does some kind of job. And if you have a hormone A, it does a little bit of a job. But there's another hormone that's permissive to hormone A. The permissive hormone isn't doing the job, but if it's present, hormone A does a much bigger job. It has a much bigger response. That's the effect we'll be talking about in greater detail in subsequent slides. Sometimes hormones are synergistic, so hormone J and K work on something. So let's say testosterone and FSH work on sperm production. They do a small job by themselves, but if you put them together, the sum of their action is greater. I'm sorry, them both working together is greater than just the sum of them working alone. That's called a synergistic effect. Sometimes hormones are antagonistic. So if hormone Y increases, then let's say that's glucagon, then blood glucose level goes up. Hormone X lowers blood glucose level. So if hormone X goes down, that would raise blood glucose level. So hormone Y could be glucagon, hormone X could be insulin. They are antagonistic because insulin raises blood glucose level, whereas glucagon, whoops, I said that backwards, I'm so sorry, so let's start again. Insulin lowers blood glucose level, whereas glucagon raises blood glucose level. And so they're doing the opposite effect, and we call that antagonistic. So what does it mean to be permissive? Hormone B is doing the job, but hormone A must be present for B to do its full job. So insulin is permissive for growth hormone to cause growth. Growth hormone is what's making you grow, but insulin needs to be present for growth hormone to do its full job. Cortisol is permissive for glucagon to raise blood glucose level. Glucagon increases your blood glucose level, but it does a better job if cortisol is present. Oh, okay. So thyroid hormone is permissive for catecholamines to increase lipolysis. Thyroid hormone doesn't break down fat. Epinephrine breaks down fat. But if thyroid hormone is present, epinephrine does a much better job. So thyroid hormone's not doing a thing to fatty acid production or breakdown of fat from lipids. Epinephrine does a little bit of a job. But if you have thyroid hormone and epinephrine, oh my goodness, it has a much larger response. So thyroid hormone is permissive for epinephrine to break down fat. Okay, so after you've viewed this presentation, why don't you take a blank piece of paper and see what you can remember about the learning objectives. Look at your learning objectives, take a blank piece of paper, write down as much as you can remember. And then and maybe do that before lecture or do that right after lecture. See how it works for you. Also work through CT 8.1. That will help you apply a lot of the information in this lecture. And then next time we'll be talking about how the hypothalamus and the pituitary interact, mostly regulating hormone secretion through a process called negative feedback. So I appreciate your attention to this video, and I'll see you in class.